Hello! In today's video, we shall learn in detail about Psilotum. By the end of this video, you will have a clear understanding about its morphology, anatomy of its aerial stem, rhizome and leaf, its different modes of reproduction, namely vegetative, asexual, and sexual reproduction, structure and development of Synangium, structure and development of Gametophyte and Sporophyte, and finally its life cycle. Let us begin with the classification and habitat of Psilotum. Psilotum is a primitive, seedless vascular plant. It belongs to the division Pteridophyta and the family Psilotaceae. Psilotum is commonly known as the Whisk fern due to the brush-like appearance. It grows mostly in tropical and subtropical regions. It can grow in the humus, example Psilotum nudum, or as an epiphyte on trees, example Psilotum flaccidum. The plant prefers shady and humid environments. Next, let us understand the morphology of Psilotum. The adult plant body of Psilotum is a sporophyte. It consists of two parts, the underground rhizome and the aerial stem. True roots and true leaves are absent. The sporophyte has a horizontal subterranean rhizome which anchors the plant to the substratum. In order to survive, it depends on a symbiotic relationship with mycorrhizal fungi that live inside the cortex. The fungi absorb nutrients for the plant, and the plant in return provides sugar. In the absence of true roots, hair-like structures on the rhizome, called rhizoids, function as organs of absorption and anchorage. The aerial stem arises from the rhizome. It may be erect as seen in Psilotum nudum, or it may be pendulous as found in Psilotum flaccidum. The aerial shoots of Psilotum are green, perennial, and dichotomously branched. It gives the plant its characteristic forked appearance. As the stem contains chlorophyll and stomata, it performs photosynthesis and facilitates gas exchange. Aerial shoots are devoid of true leaves. Instead, they bear small, scale-like, non-vascular appendages called innations. In the axils of the innations, reproductive structures are present, which are tri-lobed sporangia called synangia. Each synangium is formed by the fusion of 3 sporangia. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us learn about the anatomy of aerial stem of Psilotum. A transverse section of the aerial shoot of Psilotum shows irregular outline due to the presence of ridges and grooves. It has 3 regions: outer epidermis, middle cortex, and an inner stellar region. Epidermis is the outermost layer of the stem. It is made up of a single layer of compactly arranged elongated cells. Epidermis has a heavily cutinized outer wall which reduces water loss. Sunken stomata are present in the grooves. Each stoma has 2 dumbbell-shaped guard cells which allow gaseous exchange. Cortex is well developed and lies below the epidermis. It is differentiated into outer cortex, middle cortex, and inner cortex. Outer cortex is composed of chlorenchymatous cells containing chloroplast. It is known as the photosynthetic zone and helps in photosynthesis. It is made up of 2 to 5 layers of vertically elongated, thin-walled cells with intercellular spaces, which facilitate gas exchange. Middle cortex is made up of 4 to 5 layers of vertically elongated, closely packed sclerenchymatous cells without any intercellular spaces. It provides rigidity and mechanical support to the shoot. Inner cortex consists of closely packed, thin-walled, parenchymatous cells with no intercellular spaces. The cells of this region are rich in starch grains and primarily help in storage. Endodermis is the distinct innermost layer of the cortex. It is single-layered and made up of barrel-shaped cells. Casparian strips are present on the radial walls. They control the movement of water and minerals into the stele. Pericycle is single-layered and lies below the endodermis. Stele is the central conducting region. In the center of the stele is the pith, which is sclerenchymatous. It is surrounded by xylem and appears star-shaped. Xylem is exarch with protoxylem at the tip of each ray and metaxylem in the center. Phloem fills the space between the star-shaped arms and surrounds the xylem completely. Xylem is composed of tracheids. Vessels are absent. It conducts water and minerals upward. Phloem consists of sieve cells and phloem parenchyma. Companion cells are absent. It conducts prepared food material and transports sugar down to the rhizome. At the base of the stem, the stele is an actinostelic protostele, and in the middle portion, it is a siphonostele. Next is the internal structure of rhizome. The transverse section of the rhizome of Psilotum shows outer epidermis, middle cortex, and an inner central stellar region. Epidermis is single-layered with thin-walled cells compactly arranged. It is covered by a thin cuticle. Stomata is absent. Multicellular hair-like rhizoids appear as prolongations of the epidermal cells. Outer cortex is made up of closely packed parenchymatous cells. It shows the presence of intracellular endophytic mycorrhizal fungus, which forms a symbiotic relationship. The fungus helps the plant in absorption of nutrients. Middle cortex consists of thin-walled, loosely arranged parenchymatous cells. This layer stores abundant starch grains. Inner cortex is made up of compactly arranged parenchymatous cells. They are dark brown in color due to the presence of a chemical called flobafine. Endodermis is the innermost layer of cortex. The cells are barrel-shaped with Casparian strips on their radial walls. They regulate water and mineral transport into the stele. Pericycle is single-layered with thin-walled cells. It protects the stele and gives rise to lateral branches. Stele is protostele. Xylem is exarch, found in the center and surrounded by phloem. Pith is absent. Next is the anatomy of leaf. The leaf or appendage of Psilotum has two parts, epidermis and mesophyll. Epidermis is single-layered, cutinized, stomata are absent. Mesophyll is composed of parenchymatous, chlorophyllous cells which are not differentiated into palisade and spongy cells. As stomata and vascular traces are absent, they are not photosynthetic in function. Next, we shall study about reproduction in Psilotum. Psilotum exhibits alternation of generations. Sporophyte is dominant and green, can photosynthesize, bears sporangia, and produces haploid spores. The Gametophyte is small, non-green, and saprophytic. Sporophyte reproduce vegetatively and asexually. Sexual reproduction occurs in Gametophyte. Vegetative reproduction is with the help of some multicellular specialized propagules called Gemmae. Gemmae are small, multicellular, one cell thick, oval or pear-shaped bodies. It becomes thicker towards the center. Gemmae are formed both in the Sporophyte plant and in the Gametophyte prothallus. In Sporophyte, they are produced in large numbers on the surface of the underground creeping rhizome. The cells of the Gemmae are thin-walled and rich in starch grains. Gemmae are attached to the rhizome by a short, delicate, multicellular stalk. Each gemma contains living meristematic cells capable of continued growth. When they get detached from the rhizome, they germinate into a new sporophyte. Germination can also occur when gemmae are still attached to the rhizome. Gemmae are also produced in the subterranean prothallus of the Gametophyte. When they get detached, they give rise to a new prothallus. Fragmentation. Some species show vegetative reproduction by fragmentation. The underground dichotomously branched rhizome may break into pieces due to some physical disturbances. Each fragment contains an active growing point, due to which it can develop independently into a new plant. Asexual reproduction in Psilotum takes place by means of spores produced in trilobed sporangia referred to as Synangia. Synangia are the asexual reproductive organs. They are borne on a short stalk in the axils of small, bifid, fertile, foliar appendages on the upper portion of the dichotomously branched fertile shoot. A sporangium and its subtending fertile appendages are collectively known as sporangium complex. Development of Synangium begins in the adaxial or upper surface of a dichotomously branched axis subtended by a pair of small bifid appendages. The development of each sporangium of the Synangium is of the eusporangiate type. 3 separate sporangial primodia arise side by side. It develops from a group of superficial initial cells. Each initial cell undergoes division to form an outer primary jacket initial, which undergoes several divisions and forms 4 to 5 layered thick sporangial wall except along a line of dehiscence, and an inner primary sporogenous cell or primary archisporial cell which divides and forms a mass of sporogenous cells that differentiate into sporogenous tissue. Some of these cells develop into spore mother cells with dense granular cytoplasm. The remaining cells disintegrate into a plasmodial mass which gives nourishment to the spore mother cells. The diploid spore mother cells undergo meiosis to produce tetrad of haploid spores. The mature Synangia is a tri-lobed structure with a short stalk. It is surrounded by bilobed foliar appendages. As the three adjacent sporangia grow and mature, their cell walls fuse completely, resulting in a single Synangium. The transverse section of Synangium shows a thick multicellular synangial wall which consists of 3 to 4 layers of cells, and the outermost layer of the wall is highly cutinized to prevent desiccation. At maturity, the Synangium opens by splitting longitudinally to release the spores. There are 3 distinct locules separated by septa. Each locule is an individual sporangium. 3 sporangium fuse during development. Each locule has numerous identical haploid spores which are bilateral, kidney-shaped. Each spore is surrounded by 2 layered wall, the outer exine and an inner intine. Spores disperse and fall on a suitable substratum. Next is the development of Gametophyte. Each spore germinates exosporically. When the exine ruptures, the intine forms a small tubular outgrowth. Each spore grows into a small, underground, non-green gametophyte or prothallus. A transverse wall is formed and the whole structure becomes two-celled, giving rise to the upper apical portion and the basal spherical portion. The latter still remains within the spore wall. The apical portion divides repeatedly, forming a mass of thin-walled cellular body which becomes cylindrical, slightly branched prothallus, whose surface is covered by hair-like rhizoids. Prothallus represents the gametophyte. The gametophyte becomes infected by endophytic mycorrhizal fungus. Next is the structure of gametophyte. Gametophyte is a subterranean, cylindrical, branched, elongated structure showing the presence of an endophytic fungus in the cells. The internal structure of gametophyte shows thin-walled parenchymatous cells. The peripheral cells are heavily cutinized. Rhizoids are present on the outermost layer. Next, we shall study about sexual reproduction in Psilotum. The gametophyte is monoecious or homothallic. Both the sex organs, namely Antheridium and Archegonium, are found on the surface of the prothallus in large numbers. Antheridium is a semi-spherical structure embedded in the tissue of the gametophyte. The spherical Antheridium has a single-layered sterile jacket cells which surround a number of androcytes. It develops from a superficial cell of the prothallus called the antheridial initial. The antheridial initial divides and gives rise to an outer jacket initial, which later forms a single-layered wall of the antheridium, and an inner primary androgonial cell which divides to form a mass of androgonial cells. These androgonial cells give rise to androcytes, which undergo metamorphosis to form spirally coiled multiflagellate anthrozoids. The opercular cell of the antheridial wall disintegrates, a pore is formed, and the anthrozoids are released. Next is the structure and development of Archegonia. The Archegonium is embedded within the gametophytic tissue. It is flower-shaped with a protruding neck and a basal swollen venter. It develops from a single superficial cell of the prothallus called the archegonial initial. The archegonial initial divides to form an outer primary cover cell, which divides to form 4 neck initials that later forms the long archegonial neck. The central cell formed by the division of archegonial initial forms an upper primary neck canal cell, which gives rise to a binucleate neck canal cell, and a lower primary ventral cell, which divides to form a small ventral canal cell and a large egg. Fertilization takes place in water. A mucilaginous mass is formed by the disintegration of the neck canal cell and ventral canal cell. It absorbs water and swells. A free passage is formed for the anthrozoids to make its way into the egg. One of the male gamete fuses with the egg to form a diploid zygote. It later forms the sporophyte that remains attached to the gametophyte during early stages of development. Next is the development of embryo into young sporophyte. Zygote is the first cell of the sporophyte. It gives rise to the embryo by forming an upper epibasal cell which forms the sporophyte shoot system with rhizome, aerial green branches, and rhizoids. It also gives rise to lower hypobasal cell which develops into a foot embedded in the gametophytic tissue. Foot gives rise to haustoria-like outgrowth. It helps to absorb nutrients from the gametophyte and makes it available for the developing sporophyte. The mode of embryo development is exosporic. With the development of the embryo, the cells of the prothallus adjoining the young embryo forms a gametophytic tissue called the calyptra above the embryo. The embryo grows through the calyptra, which gets ruptured by the growing axis of the embryo. The embryo grows horizontally and develops rhizome and unicellular rhizoids. Some of these branches move upward and develop into the aerial shoot. As soon as the young sporophyte gets infected with the mycorrhizal fungus, it breaks off from the gametophyte and becomes independent. Finally, we shall look into the life cycle of Psilotum. Psilotum exhibits alternation of generations between the sporophyte and the gametophyte. The plant body is a diploid sporophyte that reproduces asexually by producing haploid spores from the Synangium. Each spore germinates exosporically and develops into a gametophyte. The gametophyte produces haploid male and female gametes that fuse to form the diploid zygote. It later forms the diploid plant body. So today, we have learnt in detail about Psilotum. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed the channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.